Okay, we're coming into the last section of lecture 25. This is pharmacodynamics. This could be a whole course in and of itself, so we're going to do just some brief highlights of things that I think are relevant and interesting. So pharmacodynamics, pharmaco, so the drug, the pharmaceutical, dynamics, what is it actually doing? What is it affecting, what is it making happen in the body? So you can think of this as what does the drug do to the body or do in the body to result in its therapeutic or recreational effect. This is different from something called pharmacokinetics, which you can kind of think of as what is the body doing to the drug. So anytime a drug comes into our body, our liver metabolizes it, our kidneys excrete it, right? All these things happen and we generally end up eliminating it from our body over time, which is why it wears off and you would have to take another dose. So we're going to talk about pharmacodynamics. A couple of general principles that I just want to put in your heads. So one is that molecules are constantly in motion, and it's this totally random motion. You might remember learning about Brownian motion. They're moving more quickly if it's warmer temperature and more slowly if it's slower temperature, but it is random. It's not intentional. But then they will also, right, second law of thermodynamics, they will tend to spread out or diffuse, right? Mother Nature doesn't want things highly concentrated in one place. They will move out. So as they're just kind of randomly, moving around, they will generally tend to diffuse, just random probability. And if they bump into a receptor for which they are specific, they will bind to it. If they bump into a reuptake protein pump, they might get reuptaked. That's not a word, but I made it one, right? So it's not as if, when acetylcholine is released into a synapse, it is like, there's my receptor, and it goes right to it, right? It's just kind of like, and some of it ends up binding. So that's important when we think about the concentration of a drug. So the more of it that's around, the more likely it might actually bind to a receptor and have an effect. Yes? Okay, so there's a little GIF that we're going to look at here. We'll see where it starts and if it works. No, it didn't work. So where's the beginning? Didn't start at the beginning. Well, so what we can see is we just have these random molecules. Oh, there was the beginning. So they start off kind of separated, these two different types of molecules. They're just randomly, stochastically moving around. And then eventually they're both going to be kind of equally distributed, generally, statistically, throughout whatever medium we are at. Okay, I can give you a headache. Another general concept that I want you to understand is the dose response curve. So what we have here, this is from the LIM textbook, is on the x-axis we have increasing doses of drug concentration, right? So scientific notation, 10 to the negative third, 10 to the negative seventh, right? 10 to the negative third is way bigger, right, than 10 to the negative seventh. So drug concentration is increasing. I have to go this way, increasing. And then we're looking at how much effect does it have on the body? How much of a response are we seeing? So if you give a really, really teeny dose down here, right, you might not see any effect, but once you kind of reach a certain threshold, then you'll start to see a response. And then as you keep giving more and more of the drug, eventually you're going to reach this kind of plateau where giving additional drug is not going to give you any additional effect. You've kind of already maxed it out for whatever reason. Things are saturated, the receptors are already all bound, whatever it might be, and giving additional drug is not going to give you any additional effect. So this is a classic dose response curve, and we're always trying to find the sweet spot where we get the optimal effect and minimal side effect, which is tricky. And the reason that's tricky is because whatever process the drug or medication is targeting, whether it be a receptor or a neurotransmitter or some other physiologic process, we use these things for many different functions in our body. So just think about acetylcholine. It's used in the synapse for the neuromuscular junction. It's used in the synapse between the pre- and postganglionic neurons for both the sympathetic and the parasympathetic nervous systems, and they have largely opposite functions. And then it's also used, released by the postganglionic neuron in the parasympathetic nervous system, right? So just that one neurotransmitter, and that's, 1, 2, 3, 4 different types of synapses that's used that we've just learned about in this class, right? And there's many, many more. So our body uses the same chemicals to do a lot of different things in different places. So anytime we're altering a neurotransmitter or altering a receptor, which is what we are often doing, we're going to have other effects aside from those that we intended, because that same process is being used elsewhere in the body. So for example, the SSRIs, the selective serotonin reuptake inhibitors, which prevent that reuptake from the synapse, therefore serotonin stays in the synapse longer and can be very useful for treating depression and anxiety disorders, can also have a lot of other side effects. So one of the very common ones actually is some GI distress, some nausea, because serotonin is involved in the nausea pathway. So if we're increasing serotonin levels, guess what? You might feel a little sick to your stomach, you might lose your appetite, or then on the other side of things, you might have an increase in appetite due to those increased levels of serotonin. So problems with sleep and sexual function, dry mouth, headache, dizziness, all of those things can come about because of the many different ways that our body will use the same neurotransmitter or the same type of receptor. So you get these other effects. In general, higher doses mean higher rates of side effects. Now, sometimes these things can be dangerous, right? In which case we would refer to it as toxicity, right? Something could be toxic, could be dangerous to you. Sometimes it's just annoying, right? Like sexual dysfunction. That's annoying. Okay, but it's not acutely dangerous. It's not going to kill you, I hope. So I also want to bring up hair loss due to chemotherapy drugs. So for example, many chemotherapy drugs work by being specifically toxic to and killing cells that are replicating themselves really, really quickly. And cancer cells, one of the hallmarks of cancer cells is that they are replicating and reproducing themselves very, very quickly, very, very fast. But they're not the only cells that do that. So it turns out that the cells in our hair follicles also reproduce themselves very, very quickly. So they also take a hit with certain types of chemotherapy drugs, which is why people lose their hair, right? So it's an unintended consequence, but not one that we didn't necessarily know was going to happen, right? It's just the same types of physiology can be seen in more than one part of the body. The other thing that I want to point out is that all cells have receptors. So we've been talking a lot about synapses because we've been talking about the nervous system. So that's pretty darn important. And so we've been talking about those receptors for neurotransmitters that are present on the postsynaptic membrane, yes? And they could be ionotropic or they could be metobotropic. But it turns out that all of our cells have receptors on them, and that is how they can respond to things like epinephrine circulating in the bloodstream or other hormones. That is how we can signal to cells things that we want them to do or ways that we want them to behave. So just keep that in mind. All cells have receptors, therefore all cells we can, give a drug or a substance that will influence the way they behave. Different cells have different types of receptors. So this diagram is showing us estrogen receptors and the types of tissues in the body that have receptors for estrogen, which we typically think of as the female sex hormone. Both male and female-bodied people have estrogen receptors in their bodies. You have them in your brain, in your cardiovascular system. If you have breasts, right, you have them in your breast tissue, your liver, your ovary, your testicles, your adipose tissue, which is your fat tissue, and your bone. Now, the other thing that this diagram is showing us, so first of all, I'm going to back up. Receptors are specific for specific ligands, right? So these cells have estrogen receptors, and only estrogen can bind generally to estrogen receptors or drugs that mimic the structure of estrogen. We'll talk about that in a minute. But then there's different subtypes of these receptors. So what they're showing you is that some of these tissues have both alpha and beta subtypes. Some have predominantly alpha, if that's the one that's red and the other one isn't, and some have predominantly beta. So it's interesting, if you're interested in drug development, is what you can do, for example, is you can try to develop a drug, maybe if you want to prevent breast cancer, right? So you can maybe develop a drug that's going to have an impact on the alpha receptors for estrogen in the breast, but then therefore, if they don't impact the beta receptors, then you wouldn't have side effects in the bone, right? So it's kind of cool. All right. Okay, I just want to remind us about the two types of receptors we've learned in this class. There are other ways of categorizing receptors. There are other types of receptors, but these are the ones that I'm going to expect you to know for the final exam. So ionotropic, those are ones where a neurotransmitter or a ligand binds, and the receptor opens, and the receptor happens to be an ion channel. So, oops, sorry, I didn't do the animation in the right order. There we go. So they are a ligand-gated ion channel. They are an ion channel that opens in response to a certain molecule binding. And then depending on what type of ion channel they are, that ion will be able to move across according to its electrochemical gradient. So both the chemical concentration and then the charge gradient will have an impact on which way it moves. Then we have the metabotropic, otherwise known as G protein coupled receptors, because they have a G protein that is attached or coupled to them. And what that is going to do is then trigger a cascade of actions via what's called a second messenger, right? So it's like, okay, you gave me the message, and I'm going to pass it to this other guy, and he's going to go take it to the next thing. And that second messenger could end up opening ion channels, or it can cause all types of other effects in the cell. It can impact what regions of our DNA we transcribe and activate, what types of proteins we're making in the cells, all kinds of things. So metabotropic receptors can have lots of different impacts depending on what's happening. Okay, just like there are different types of receptors, there are different types of ligand. Remember, a ligand is a molecule that will bind to its specific receptor. So the ligand, you can think of it as, so these are all ligands, but like you can think of like your endogenous or the natural ligand, right? So acetylcholine, for example, it is a ligand for acetylcholine receptors, yes? But then if we're talking about drugs or other things, we can categorize them based on their actions and behaviors at the receptors. So they might be agonists that promote the activity that is normally induced by the ligand at the receptor. They could be antagonists, right? An antagonist is like an enemy, somebody who fights you, who prevent the ligand from doing what it would normally do. Or they can be allosteric modulators, which sounds very sophisticated if you ask me. Let's talk about agonists first. So agonists behave, they almost mimic like the kind of natural ligand. For example, I was talking about acetylcholine. Well, it just so happens that some of the acetylcholines, which are called the nicotinic acetylcholine receptors, we learned about those before, the natures, nicotine can also bind to them. So this diagram had so many errors in it, but I still liked it, and I just blocked out a lot of the errors. So either acetylcholine or nicotine can bind to this receptor, which is an ionotropic receptor, and it will open and allow sodium ions to travel into the cell according to its electrochemical gradient, which would move it closer to threshold, right? It's going to depolarize the cell. So nicotine is an agonist. It acts like it mimics the kind of natural ligand for the receptor. So nicotine is an acetylcholine receptor agonist. It activates it, makes it do the thing that it would normally do. Morphine, for example, binds to opioid receptors. That shouldn't be surprising. So we have natural opioid receptors because we have natural opioids that get produced in our bodies, and they tend to be metabotropic, and morphine will bind them and activate them the same way that the endogenous opioids would, actually a bit stronger. Okay, so in each case, the receptor is kind of almost being tricked. It's acting as if it's the ligand it normally expects that we would have within our body, right? Whether that be acetylcholine in the case of nicotine or endogenous opioids in the case of morphine. So it's a mimic. It is activating the receptor the way the normal thing that binds would. Some agonists are what we would call full agonists, meaning they can do the whole thing. They can activate it just as fully as we would expect, as it can have activated all the way, and some are partial. So obviously they can only activate it partially, even if we give higher and higher doses. So that's what we're seeing here in the diagram. So here in green is our full agonist. It's going to bind to the receptor. Here's the binding site here. It's going to attach, and that's going to result in a full response. So dose response curve, right? We're going to get just as high of a response as we would with kind of the natural ligand. A partial agonist, is still going to bind to that same spot on the receptor, it's not going to be able to activate it as fully. So you're not going to get as big a response as you would from a full agonist. Full versus partial agonist. There we go. Now let's talk about the antagonist, right? So they're fighting you. They're not letting you do what you would normally do. So what an antagonist does is it binds to the receptor. So it's going to bind to the receptor here and basically shuts it down. And it can shut it down in a variety of different ways, but the key thing here is it prevents the receptor from doing its thing. Whether it is an ion channel or whether it is a G protein coupled receptor, it's just like, nope, we're shutting you down. So that's what an antagonist does. One type of antagonist is what's called a competitive antagonist. And a competitive antagonist is going to specifically bind to the same spot that the natural ligand or agonist normally would. So that's actually what's shown in this diagram here. They're showing it would bind to the same spot. And then it prevents the natural ligand or agonist from being able to dock and land on that receptor. Therefore, it can't trigger the response. That's a competitive antagonist. I think of it like when you're playing musical chairs, right? And only like so many people can sit, and if somebody else gets to the chair before you, like you're out of luck, right? So that's what a competitive antagonist is, right? It gets in there, and then the regular agonist, the regular ligand can't bind, and so you can't activate the receptor. This is another image of that. So here's your normal agonist, whether that be a drug or the endogenous, mean the natural body thing. It would normally bind and open the channel. This one happens to be an ion channel, it looks like. But if the competitive agonist, which you see here, binds to that spot, then the agonist can't. And it can't bind, and the competitive agonist doesn't open the channel. And so now it's like, ha, 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 I got here, and now you can't. And so the channel won't open, or the G protein complex won't activate, and so you will not activate the receptor. You won't be able to. So what's cool about this is Narcan is a competitive antagonist. So we're going to back up a little bit, and opioid drugs like heroin or fentanyl or Percocet or whatever it may be, they are opioid receptor agonists. So they are drugs that bind to the opioid receptor and activate it. That's why it feels really good to take those drugs, so I'm told, right? So in an overdose, it just so happens that they can suppress your respiratory center in the medulla and the pons, nice little review there, and shut down your breathing. That's a bad thing, right? So naloxone or Narcan, which we use to reverse an overdose, is a competitive antagonist. So what we see over here is they've made this little graphic that is an opioid receptor in the brain, and here's your opioid, right, your morphine or whatever it might be, and it binds to the receptor and causes the effect. In this case, stopping you breathing. So it turns out that naloxone, which is Narcan, is a competitive antagonist. So it's going to be like, oh, no, excuse me, I'm going to take that spot. And when I take that spot, it doesn't activate the receptor. It just blocks it. And so now the opioid molecule can't bind to it. It can't activate the receptor, and we can't get the drug effect, which in this case was killing you. So what's interesting about Narcan or naloxone is it's not only a competitive agonist, but it has what we call higher affinity for the receptor. So it's much better at binding to the receptor than the opioid molecule is. So it'll basically kind of kick it out. So it's like it's really competitive game of musical chairs, and it's actually kicking you out of the chair that you were already sitting in. So that's why it's so effective. So Narcan, interestingly, is available as a nasal spray. So that insufflation, we're getting it through the mucous membrane right in there in the nose and going into the bloodstream. Or you can give it as an injection. Usually IM is preferred over sub-Q because it's more rapid, but the over-the-counter kind is usually the nasal spray. And so you administer it, right? And it goes through the bloodstream and up into the brain. And then it is going to bind to the opioid receptor and kick the opioid off and prevent the opioid from being able to bind. Therefore, the receptor is no longer being activated, and the person wakes up and starts breathing again. It's amazing. It is a miracle. It is fabulous. Yay, competitive antagonists with higher affinity for binding. Now, I will tell you, sometimes depending on how much of a dose of an opioid somebody has had in their overdose, sometimes you have to give a second dose of Narcan. So if after a couple of minutes, and hopefully you've been doing some rescue breathing in the meantime, it hasn't worked, you can give a second dose. Now, also keep in mind, Narcan wears off after about 30 to 90 minutes. A lot of opioids last longer than that, so it is very possible that when the Narcan wears off, they will stop breathing again. They will lose consciousness and stop breathing again. So for that reason, anyone that is treated with naloxone to reverse an opioid overdose needs to go into medical observation. They need to go to the emergency room. We need to keep an eye on them because they might slip back into overdose again. Okay, so we've now talked about agonist and antagonist. Now it's time for the fancy allosteric modulators. Here we go. The word allosteric comes from the Greek, so allo means other or different than, and steric refers to place, so it's an other place. So if we look at this diagram here, we can see the orthosteric site over here, and then the allosteric site over here. So the orthosteric site, ortho means like straight or correct. So like an orthopedist will straighten your foot if it's broken. That's kind of where that comes from. So the normal binding site is the orthosteric site, but allosteric modulators is like, no, I'm fancy, I'm special, I'm gonna bind somewhere else on the receptor. So they bind to this allosteric site, right? They can't just do the typical thing. So they're going to bind elsewhere, and they do not directly inhibit or activate anything. What they do is they modulate the response of the receptor should the regular agonist or ligand bind. So here what we see is if it's the agonist alone, so this is kind of this first one here, agonist alone, so it's going to bind to the orthosteric regular site, and we're going to get the typical response. Now, some allosteric modulators will increase responsiveness of the receptor, and so those are called positive allosteric modulators, known as PAMs. So if I have a PAM, which is going to modulate to make my receptor more responsive, I'm going to have an enhanced response. I'm going to have a bigger response. Whereas if my allosteric modulator is inhibiting the response or decreasing this responsiveness, that would be a negative allosteric modulator or a NAM. And so in this case, so that's this one here, I have a NAM, so it's going to negatively influence responsiveness. So now I'm going to have a smaller response than I had at baseline over here. Now it's important to note that allosteric modulators just modulate the response to the regular agonist or ligand. They do not activate or deactivate the receptor on their own. They just change how it's going to respond to the actual ligand or agonist. So Valium, interestingly, which is in the same family as it is diazepam. It's in the same family as Xanax. All those Klonopin, Ativan, lorazepam, those are all benzodiazepines, and all of the benzodiazepines are positive allosteric modulators. Okay, so let's take a look at this. I gotta put my glasses on. Okay, so what we're looking at here is a GABA receptor right here. This is a GABA receptor, and it's spanning the cell membrane. Right here's the cell membrane. This blue circle is meant to represent GABA. And when GABA binds to its receptor, this would be its orthosteric receptor, right? The usual regular receptor binding location. When it binds, then it allows, look, chloride ions. Chloride ions move into the cell. Now, chloride ions are negative. They're going to hyperpolarize the cell. They're going to make it more negative. They're going to move it farther away from threshold. So that is an inhibitory postsynaptic potential. It makes the neuron less likely to fire. So at baseline, what GABA does at this particular receptor, it's different at different receptors, but most receptors, what GABA does is makes the neuron less likely to fire. So it's decreasing neural activity. That's what GABA generally does. So we often refer to it as an inhibitory neurotransmitter because it decreases the activity of the neurons, decreases the likelihood that they're going to reach threshold and fire an action potential. So that's at baseline. Now we're going to add benzodiazepine, which is this little teal thing. Now notice, it binds to the receptor at a different place. Allosteric site, right? So it's special. It has to bind to the separate site. And when it does that, and we also have GABA released into the synapse, right? In the presence of GABA, when a benzodiazepine is present, more chloride ion, right? So this is a positive allosteric modulator. It's going to make the receptor do its thing bigger, and you're going to get more chloride into the cell. You're going to get more hyperpolarization, so you're going to have even further decreased likelihood of threshold and action potential. Therefore, you're decreasing overall electrical activity in the brain. So that's why Valium mellows you out, and right? Because it's just like shutting down the brain because it is enhancing the effect of GABA, which is to make a neuron less likely to fire. A little complicated, I know, but also very cool. So in summary, our molecules behave stochastically. They're randomly bopping about, right? There's this dose response curve where in general, the more drug, you have to give a certain level of drug to see an effect, and then you give more and more, you see more and more effect, and then it usually kind of levels off as you reach maximum effect. Side effects occur because these systems are used in all different parts of our bodies and different systems in our brain. So if you impact one part of it, you often impact other parts of it. Let's see, then all cells have receptors, which is how we can have drug effects on all different types of cells. Remember, they might be ionotropic, they might be ligand-gated ion channels that open and let an ion through, like the GABA receptor that we just saw that lets chloride ions through, or metabotropic, where they are G protein coupled, which activates a second messenger cascade that can cause a variety of things to happen. So ligands can be of 3 different flavors for our purposes. So agonists are going to increase the activity of the receptor. They kind of mimic the natural ligand and they activate the receptor and make it do its thing. If they're full, they can do it all the way. If they're partial, they can only do it partially. I love when things make sense. Antagonists are going to kind of fight and prevent the natural ligand or a drug agonist from being able to do its thing and activate the receptor. If it's a competitive antagonist, that means that it binds to that orthosteric site, that site that the agonist would normally want to bind to, and then it's like, ha ha ha, you can't bind here, and therefore prevents it from being able to activate the receptor. So Narcan was our example for that. And then lastly, the very fancy allosteric modulators, they bind to a different site because they're different than everybody else. So that's what allosteric means. And they're going to modulate how the receptor responds to an agonist, be that the kind of the natural ligand or a drug agonist. And so they might increase its responsiveness, in which case it's a positive allosteric modulator or a PAM, or it could decrease its responsiveness, in which case it is a negative allosteric modulator or a NAM. In the instance of Valium and other benzodiazepines, they are a PAM. So they attach to the allosteric site on the GABA receptors that make it so that when GABA binds, you get even more flow of those chloride ions into the cell, even more inhibitory postsynaptic potentials, hyperpolarizes you, takes you further away from threshold, and decreases neuronal firing. And that's how they work. Pretty cool. All right, that was a long one. Thanks for sticking with me, everybody. That's it for lecture 25.